Hi, I'm Carrie Lebowski. I'm a professor of soil science and a soil fertility and nutrient management extension specialist. In this last segment for the soil fertility, we're going to be discussing the uses and limitations of plant analysis. So first of all, we need to talk about there's different types of plant analysis and make sure we're all on the same page. There's cell sap tests. Now these are typically in-field quick tests. They're semi-quantitative. That's not what we're going to be talking about for the bulk of this presentation. We're going to be talking about total analysis. These are lab tests where a whole plant or a specific part of the plant has been sampled and sent to a lab for analysis. These provide an indicator of nutritional status and assumes, and this is a big assumption, that the nutritional status is related to the soil nutrient availability. And there can be reasons why it may not be. Now there are several uses for plant analysis. One is to help aid in the identification of deficiency symptoms. So we can, say have a field of soybean that we think maybe is potassium deficient, so deficient is showing some symptomology, but we're not really sure. Tissue testing can help us with that. It's also useful in helping us determine the nutrient supplying capacity of the soil. In order to do this, you need to have some soil tests and some field history, and you also need to ask some questions like, is compaction affecting nutrient uptake? In research, it has more uses, I think, in that it helps us look at the effect of nutrient additions, whether it's fertilizer or manures, how that affects not just yield, but how is that affecting nutrient composition within the plant. And so it can be used in studying the relationship between the nutrient status of the plant and crop performance. Now, there's some symptomologies of nutrient deficiency. For example, potassium. We often see that on the leaf margins as a yellowing. But it's important to keep in mind that the symptomology is not necessarily a direct effect of the deficiency. It's caused by a buildup or lack of intermediary compounds in the biochemical pathways of the plant that results in nutrient deficiencies or unavailability. Symptoms may be from multiple nutrient deficiencies, and those are hard to tell what's happening when you've got, say, two nutrients that are deficient and how that displays on the plant. And deficiencies of one nutrient could often be related to excess quantities of another nutrient. Okay, so that gets a little goofy in looking at some of this. Symptoms might be similar to chemical damage or insect feeding. That's really important. Before you start going down the road of looking at nutrient deficiencies and plant analysis, probably need to be thinking about what's been happening in that field and the management history. Is that insect damage? Is there herbicide injury? And then several factors can produce the same kind of symptomology. A lot of micronutrients can produce, interveinal chlorosis, and sometimes those are a little hard to tell visually what's going on, and so plant analysis can be helpful in that regard. Now there's some components to plant analysis, and so part of it is what to sample and when to sample. Okay, and what's extremely important here is that the correct plant part must be sampled at the correct growth stage to even attempt interpreting the results. If you haven't sampled the right part at the right growth stage, any type of interpretations that we have are not going to be useful. So if we look at some of our more common crops here in Wisconsin, we have, so for example, alfalfa, we've got the growth stage, bud to first flower. You want to sample the top 6 inches, and ideally you want to take that from 30 to 40 plants. Corn, we have a lot of opportunities here. We can do some sampling prior to the crop being 12 inches tall. You do a whole plant sample. Pre-tasseling, you do the leaf below the whorl, and that would be the most leaf with the most fully developed leaf, meaning the leaf collar is exposed. And then with corn, you take tassel to silk, you take the ear leaf. Again, the collar has to be exposed. So you have some different things. Very important to note with soybean. prior to or at initial flower. So this is R1. That's when we have interpretations for that. There are a lot of soybeans that are sampled much later in the growing season, and it's very hard to compare, and perhaps near impossible to compare results from samples taken at an R4 soybean with criteria that were developed on an R1 soybean leaflet and petiole. So remember, correct plant part for the correct growth stage. We need to, and here's the reason why for that. If we look at this graph, we've got time here in the growing season. We have early growing season, mid-season, and late season, and the nutrient concentration in tissue. And typically, we can see that tissue nutrient concentrations decline as the growing season progresses. And this makes sense to some extent because we've had nutrient uptake, but then as the plant grows, it's getting bigger. We're getting more biomass, so we're getting some dilution. And so the critical nutrient range, or the sufficiency range for plant analysis would be in this area in the middle called the CNR, the critical nutrient range. Above that range, you have adequate nutrients. Below that range, they're deficient. And in here, it's a little bit tough to tell what's going on. And when we think about sample handling, this is really important in how we handle the samples. And I suspect that many samples are probably not handled properly based on some of the questions that I get once analyses are back. Don't use plastic bags, paper bags. You need it to breathe. If soil has splashed up onto the plant, and this is pretty common when you're sampling small plants early in the season, you just get raindrop splash. You want to brush off that soil. Don't wash it off because you could potentially pull nutrients out of the plant. So you want to brush it off, and you don't want to leave it on there because then you get some false readings where you get really high iron values, and some of your micronutrients look really messed up, and it's because there's soil that was on the plant. And so this test in the lab for tissue analysis is not just, it doesn't just get what's in the plant. If it's got soil on the plant and the whole thing goes in the grinder, it's not going to know where it comes from. So that's why you got to brush it off. You need to clearly label your samples as with anything else, and fill out the submission forms completely because that does help in the interpretations, especially with a crop like corn where you've got interpretations for a few different growth stages. And I think it's really important that before you take a sample, you know what lab you are going to send the sample to, and you've talked to them in advance to make sure that they may have more specific information or manners or methods of how they want you to handle the sample and submit it. And so following their protocols to make sure you get a good result is really important. Now with plant analysis interpretation, there's kind of three main methods. The sufficiency ranges, it's also considered the critical nutrient concentration. Then we have the DRIS approach, that's the Diagnostic and Recommendation Integration System or Integrated System. And then PASS, the plant analysis with standardized scores. Now the sufficiency ranges we can think of them being a little bit more like soil test values. You just look at a parts per million or a percent, and are you too high, just right, or too little? The DRIS approach and the PASS approach look at ratios between some of the nutrients, and we'll get into that in more depth. But it's important to note that DRIS scores have not been developed for all crops, and PASS scores also have not been developed for all crops. And when we get into this bit about interpreting plant analysis, we have to keep in mind that different regions let's just focus on the U.S., just different regions within the U.S. There's different growing conditions, the soils are different, to just take sufficiency ranges or DRIS scores from another part of the U.S., say the Southeast, might not produce appropriate interpretation for us here in Wisconsin. So there does need to be a certain amount of site-specific calibration because we can see differences between location. There are also some differences between hybrids. I've observed that myself. When we look at corn hybrids or soybean varieties grown at the same location, there are some differences there as well. So it's not a solid science. There's a lot of wiggle on this type of approach. So the sufficiency range, this graph kind of describes it nicely, I think, where we have our concentration of nutrient in the crop tissue. Remember, this is a dry matter basis, so it's going to be parts per million or percent depending on which nutrient. And this is the growth as a percentage of maximum growth. And then we have our red line, so this is that relationship between percentage of maximum growth and the tissue concentration. And we have, the line goes up, it's pretty steep, it bends over and then flattens off. And so when we are, here I have it drawn at 80% of maximum growth, that's considered definitely deficient. At 80 to about 100% of maximum growth, that's the critical nutrient range. There often are no symptoms, visual symptoms in this range, so it may be difficult to know that you're not getting maximum growth in this range. Then when we hit 100% of maximum growth, this is more in the luxury consumption range. The tissue nutrient concentrations may increase, but the plant doesn't need more of it. So it's just consuming it because it's just taking those nutrients in passively. And then we get to a point where we may see toxicities. So the tissue concentrations get so high, we start seeing growth go down. And often there's visual symptomology associated with that as well. Now, 10% reduction in yield, that's often where a critical concentration is placed. Again, these are not perfect numbers. We have differences between hybrids, differences between locations. And I'm going to take the time now to point out that in Wisconsin and most other states that I'm aware of, a lot of the plant analysis research was done in the 1980s, some in the 70s, and it trickled into the early 90s. There's been very little research in this area since probably about 92 or 93. A few of us have started working on this again, but it's important to keep in mind that a lot of this work was done on pre-GMO crops and also crops that, corn in 1990 was yielding what compared to corn today. So we have a lot more biomass out there, and that can be affecting how the interpretation of these nutrients. So keep all those factors in mind, that this is a less precise science, but it can be a useful tool. So if we get into these sufficiency ranges, this is an example. This is a piece of one of our plant analysis reports from our UW labs. We have over here on the left the plant results. So we've got the element, and then whether it's considered low, sufficient, or high. And in this example, this alfalfa tissue sample was considered low in sulfur, sufficient in all other nutrients. This particular sample was submitted with analogous soil sample as well, which is what I really suggest for plant analysis when you're trying to diagnose what's going on, is you have a plant plus soil. And so this is just the results of the soil test in that area. And you can see that sulfur was considered low on the soil test, and P and boron were high, potassium was in the optimum range. And then we've got this bullseye chart over here that everybody seems to love, but all it is it's just taking these results here from the plant analysis and the soil analysis and putting it into this bullseye. So how you look at it, the black dots represent the nutrient concentration. So here's the nutrients as spokes, and you have your nutrient concentrations for the different nutrients, so they move in and out. And if you're in the center area, that would be considered low. The darker green is the sufficient range, and then we have the outer ring would be considered high. So here in this example, you can see all the nutrients are in the sufficient range except for sulfur, which is low. And that's exactly what that table is telling us. Now the other thing it does here is it puts these yellow dots are the soil test results. So soil test boron, potassium, pH. So you can see where they're at. Are they low, sufficient, or high? And then we have a red marker here. It's noted that's our target pH for the crop. So this is saying that our pH of the soil was actually a little higher than our target pH. Not necessarily harmful, but just something that's there. So that's how you would look at this. And we're looking to, where are we sitting? Are we sufficient, low, or high? Fairly straightforward. I think that's why people like these sufficiency ranges, is they are a little straightforward, but they can be influenced by a number of things like we discussed. Growth stage, plant part, where you are, and hybrid. So you need to keep that in mind. That leads us to the DRIS approach. So there was a number of these limitations to the sufficiency level that can affect, how you would do the interpretation. So some folks decided they'd try to do something better. And so they developed this DRIS approach. And the idea is that you don't just look at the individual nutrients for their sufficiency, you evaluate combinations of nutrients, essentially ratios. And you then combine these ratios mathematically to provide an index. And the ideal index value or ratio is 0. And so the more negative the index, the greater potential you have for deficiency. And the opposite, the more positive, the greater the potential for toxicity. And when we look at an index value, anything between -15 and +25 is okay. So you don't have to have a 0 to be considered okay. There's wiggle here. So -15 to +25 is okay. Now we in Wisconsin, we have DRIS norms for a few crops that are listed here. Now as people began working with DRIS a little bit more, so this was really to get rid of some of these limitations of hybrids and locations and all of that. But they found that it didn't fully eliminate all of that. There were still some problems with DRIS. It wasn't as perfect as what it could be. So here in Wisconsin, Emmett Schulte and John Baldock, they developed this PASS, the Plant Analysis with Standardized Score. This was developed right before Emmett had retired and John right around the time he left the university. And it really hasn't gone much of anywhere in that time because it was also occurring at the time when people were generally not conducting plant analysis research anymore. So the idea behind the PASS is that it's this hybrid system. You combine these independent nutrient indices with dependent nutrient indices. And the PASS independent nutrient indice is similar to the sufficiency range. So the idea is that you're going to take the good of the sufficiency range and the good of the DRIS and put them together and not have any bad. Okay, that was the idea. And so these INI values, they range between -10 and +10, and they're considered sufficient. If you have an INI value less than -10, that's considered deficient. Now, nutrients in the PASS system are divided into two groups. Yield response is common on the reports. Let's back this up. On the reports, the yield response is common in the dark green areas that are bolded. And areas where yield response is rare, that would be the light green values on the report. So it's just kind of saying, do you expect that you're going to get a response or not? Now the PASS dependent nutrient indices are similar to the DRIS values. Okay, they're looking at ratios, and again, as I said before, the idea behind PASS is to blend the good of both sufficiency and DRIS. Now we have at UW PASS norms for alfalfa, corn, and soybean, and nothing else. And I'm not aware of anybody else that's using this system. As I said before, the sufficiency ranges it's a more straightforward approach. There are problems with it, but it's a little more straightforward. And I think that's why people like to use it. DRIS and PASS are a little more complicated to look at. And here's an example of DRIS and PASS scores at the bottom of our plant analysis reports. We've got DRIS and PASS. We've got it broken up into yield response is almost certain, it's possible, remote, or unlikely. And there's some footnotes at the bottom of these reports that you can read. So it's kind of nice that you can look at them and kind of decide and how you'd look at the whole package of what's put out is you take, okay, what does the sufficiency range tell me? Am I sufficient, high, low? Does that match up with DRIS and Pest? And sometimes all three of them kind of tell you the same story. Yeah, sulfur's deficient. Because remember that previous, for the sufficiency range for that alfalfa, it was sulfur was deficient. And this is the same report, and both DRIS and Pest are coming up with that. Sometimes it's straightforward like that. Other times we might see where DRIS or Pest either brings in another nutrient or rules something out that the sufficiency range didn't. So you need to think a little bit more. But again, having soil samples and knowing field history are really important in doing these types of interpretations. So that gets me to some of our limitations to plant analysis. So one of the big ones is we might be able to determine that there's a deficiency, but can we correct it in time? Okay, so it's possible that we might have determined the deficiency, but it's already caused yield loss. We can't recoup the yield loss. It's possible that the crop might not respond at the growth stage it's currently at. Another bigger problem is often the crop may be too large for nutrient application. Now, more co-ops are getting large high clearance equipment. There's more opportunities than there used to be, but it still might just not be able to do anything about it. And we can't always rule out the fact that sometimes the weather is not favorable for conducting an in-season application. So there's a lot of reasons with plant analysis that we may be able to kind of figure something out, but we can't do anything about it for this crop. It's just a done deal. But it might help us in thinking about, is this an issue that we're potentially going to have in the future on this field for other crops, and then to do something about it for the future. And so it's important to keep in mind that plant analysis is not a clear-cut tool. That's why I've got this in super big font, in bold and underline. It is not a clear-cut tool. You should not consider going into a field that looks beautiful, taking samples and running them through the lab and saying, oh yeah, that looks boron deficient. And then coming back at the end of the growing season and having the best yields ever on that field with a fully filled out ear of corn. Boron deficiency, the tip does not get filled out. So those types of things can happen. You get incorrect interpretations when you just go into a field that looks good. The searching for the hidden hunger aspect of it. Plant analysis is not as good at doing that. It's billed that way at times, and you can even find older textbooks that suggest that, but in practicality it's not very good. What it is good at is if you have a problem in the field. Particularly if you can see that there's something going on in the field, there's looks like something looks better in one area than the rest of the field, or there's a poor area. Then you can use it to help diagnose the the problem. But when you're doing that, you need to collect all of the evidence. You have to be a sleuth here. So one of the things you need to do first off, and kind of alluded to this before, you need to rule out other problems. Herbicide injury, that's a key one, and insect issues. If you can get those ruled out, then you can start focusing on the nutrient aspect. What kind of symptomology is out in the field. Get out the shovel and dig some roots. What does the root system look like? Do you have a flat, pancakey kind of root system with thick, zigzaggy type roots that suggest that you've got a whole bunch of compaction out there? Maybe the problem isn't really nutrient availability in that you don't have enough nutrients there. It's really the compaction is causing and limiting your nutrient availability. You need to look at the weather prior to these deficiency symptoms showing up. And sometimes it's not just the couple days before. What has been the growing season weather as well. Current field conditions, field history is really important. And then again, the tissue and soil analysis combined. Those can really help diagnose problems. And there are plenty of times where we were never fully certain what was wrong in the with a particular patch in the field because none of it was quite conclusive. You can maybe go in and if we think it was something, maybe apply some nutrients, but sometimes it's not really possible because it's too late in the season to do much. So again, plant analysis is a good diagnostic tool for when we have problems. Not such a good tool for trying to correct a hidden hunger that may or may not exist. And so that brings me to examples of where good uses of plant analysis, as I just suggested. Here we've got some K deficiency on soybean. So maybe you're looking at this and, yeah, I think it's K, but maybe it's not. If you take samples from this plant and then you've got another plant that looks good and you take samples from that, maybe you have some soil samples from both areas, that would really be a good diagnostic. Yep, it's K. Oh, we got a lot more K in the green plant, and we get yellow leaf margins, a lot less K. Yep, that would do some confirming because you have some comparison. Here's another example on the right. You'll see this again when we talk about pH and liming, or you saw this. This was an area where the soil pH was really low. And when you do plant analysis in an area like this with these really poor growing plants and then you go outside of it, there's all kinds of strange things that show up in the plant analysis that are very hard to interpret, that you need the soil, and then you say, oh, yeah, that soil pH is really low where there's no crop growing, and it's okay where it's not. So it's really part of a diagnostic package. And with that, we don't have a lot of resources for plant analysis. There's a Wisconsin crop management paper from a few years ago that I put together that essentially outlines what we just talked about today.